So my name is David Abura. I'm a director of Cordio East Africa, and we're a research and conservation organization based in Mombasa in Kenya, working on coral reefs in the Western Indian Ocean. Cordio is based in Mombasa in Kenya, and all of our staff have on the whole been based there. And here we work with the Kenya Wildlife Service, the Marine Parks Organization. We also work with fishing communities and researchers as well. And throughout the other countries of the region, we always partner with similar organizations and experts in those countries. So researchers, park rangers and wardens, and then also communities and small-scale fishers who have interest in the reef and who work with us to monitor the reefs where they work and where they live.
My name's Melita Samoilis. I'm a director of Cordio East Africa. We're going to be talking about this particular coral reef monitoring protocol, which is more for scientists. But we've also developed a modified one based on the same sort of principles for communities. It's for communities, coastal communities, to be able to monitor their own coral reefs, for example, in locally managed marine areas of reef that they're managing for themselves and with government.
So we have done a lot of training. We've set up this standardized protocol for the region based on what's being done in coral reefs in other parts of the world. We work very much through the Global Coral Reef Monitoring Network. That's a global entity that standardizes monitoring around the world. We help run that system in the Western Indian Ocean in our region. And then we also work out how to tailor it to the different needs of different stakeholders. So researchers will have more detailed and high-end needs for monitoring. Marine parks can do with slightly less detailed data, but they need it across the marine parks and for rangers to be able to do that. Then also communities need to monitor the areas that they fish, that they manage, and they may need slightly different monitoring methods as well.
If we don't monitor, we don't have any evidence. We need that monitoring data as evidence. Why do we need evidence? Not just for the scientists, we need it for other stakeholders. We need it for the general public. We need it for coastal communities. We need it for government. Because everyone wants to see what effect your different interventions are having. Whether it's a marine park or it's a new fisheries management legislation, the monitoring data will tell you what impact it's having. We need to standardise monitoring because what we really need is that everybody's using the same tools, the same methods, which really means the same indicators, the same metrics. So whether it's benthic or whether it's fish, we want lots of people monitoring all over the place at the same time. And when you have that, you can bring that data together, and it's much more powerful than just doing a little bit of concerted monitoring in one place. But we definitely want to work with as many other monitoring teams as possible, all using the same method, and then the data's comparable.
In monitoring coral reefs, we have to understand what's happening with the dominant parts of the ecosystem. So that's the corals and the algae on the bottom, and also invertebrates on the benthos, we call it. So our monitoring protocols really focus on identifying these main species or types of organisms that cover the bottom. Then we also monitor the fish as well, because they are dominant consumers on the reef. There are herbivores and predators and piscivores that all sort of control the ecosystem. And then there are other groups of organisms like sea urchins and so on, and it depends on the needs locally if those are monitored or not, if they're an impact. Some predators like crown of thorns as well can be these are a sea star or a starfish that eats corals. These can be very dominant in some places, and so some programs monitor those as well. So with a focus on the benthos, the benthic indicators, and the corals in particular, the most basic monitoring we do is just to monitor coral cover, the total cover of hard corals on the bottom. But then to understand better what's happening with those, we can distinguish different functional groups of corals, but also different genera as well. So we know the composition of the coral community. And that tells us a lot about the rate at which they grow, their resistance to high temperatures and coral bleaching and so on. and the recovery potential of the reef. Then we also have a protocol for looking at the size classes of corals, so we have a sense of whether the reefs are dominated by small corals or large corals, and then very importantly also new corals, so the tiny recruits or the juvenile or baby corals that come in from the water column and settle on the reef, and these are very important for recovery after massive bleaching and mortality events especially.
You know, you look at all the fish on a reef and you go, which ones should we count? You can't count them all. So we selected the families that we believe, and this is based on science done globally on coral reefs, indicator families that we believe really tell us something about the state of the reef. I'll give you some examples. Butterfly fish, which are easy to monitor as well. They're very easy to identify. They're a great family to monitor. So if you can't do anything but one family, do the butterfly fish, because they're very closely related to the state of live coral. That's one. Then other families like snappers, like grouper, they're important in fisheries. They're good food fish. So we need to monitor them as well to see what impact fishing is having or what impact fisheries management is having. And then there are others that are more nuanced, have different roles, particularly in the herbivorous fishes such as the parrotfish and the surgeonfish. So we selected in our standard protocol a range of families, there are 11, that span those different indicator roles, okay? And they also span different roles in terms of their ecosystem function, because we wanted to have a really good representation of what we're measuring in terms of the fish community. So species that are, maybe they're just eating detritus versus species at the top of the food chain that eat other fish, like groupers, they're piscivores, and everything in between. So we've got a range of those.
Site selection is an important part in coral reef monitoring. You need to know your site that you want to do monitoring. The site selection could be guided by the objective of undertaking monitoring. Could be maybe undertaking baseline survey. This means you have your target sites that you want to put interventions, so that will be your site. Or if you have common monitoring sites which you have an annual program as an example of doing monitoring, so that will be your site. It's also important that the site to be in a coral reef, as that is the target for the monitoring. So after you have selected your target site, then you need to follow the normal protocol of planning your dive. And then when you leave ready to do monitoring, when you reach at your site, the first thing is to record the site information, which sometimes we term it metadata. So that includes the name of the site and the station, the location that the site is located, and the coordinates, that is latitude and longitude. Firstly, benthic survey, it gives you the cover and show the balance between hard coral cover with other benthic groups on the reef. The first thing you need is a transect. So this is the first step. The same procedure you use in selecting your site, the same you'll use when you're underwater to select the reef where you will lay your transect. So you start laying your transect by tying the first end of your transect to a rock or a coral, the head of the coral, but you should avoid breaking the coral. And then the person laying the transect will pull the transect and make sure the transect runs in a straight line and parallel to the reef, either with the same depth or the reef crest or the back reef, and make sure at certain distance tide the transect so that it become tight. And the number of transects also depend on the reef. In some reefs, you will need 2 transects if the reef is long. Benthic surveys has many methods that are being used globally and also in the region. But 3 most methods are being used and have been explained in this guide, and their data are comparable. These methods are line intercept transect, point intercept transect, and photo quadrants. Any monitoring program could choose any of the 3 methods. For line intercept transect, you need to have a transect and a slate and a datasheet. A datasheet could be printed in an underwater paper or could be written on a slate. The observers you need in this method are 2. One who will be recording and the other one will be laying the transect. Depending on your choice, you can have a 10-meter long transect or 20-meter long transect. The recording in this method, you do record based on the change, the distance change by the benthic categories. This means as you start from the 0 point, you record the benthic categories in that distance to the point which that category ends. An example, 2 centimeter. Then you record 2 centimeter and the benthic category in that distance. And you continue the same into the meter, 10 meter or 20 meter based on your selection. The minimum number of transects you can use in this method could be 6. The second method is point intercept transect. This is one of the easy method, and it is the same method that is being used by the low-level program, like the community. So this one also could use a 50-meter transect, and then you record a benthic group at the point of your choice. Could be after every 20 centimeter or 25 centimeter or 50 centimeter. Also, the length of the transect could differ based on your selection. The recording is done at each point. So if, for example, it's after 20 centimeter, it means you record from the 0 point, then you go to 20 centimeter point, you check beneath that point what is the benthic category in that point. And then you move to 40 centimeter, you continue with the same sequence up to the last point or at the end of the transect. And we also have a photo quadrant method. As the name suggested, this you need a camera, which could be an underwater camera or a normal camera with underwater case. So this method, generally you do take photos randomly across your or along your reef. But to avoid taking more than one photo at the same point, you may need a guide, which we use a transect. We lay 25-meter transect, and then you take photo after every 3 to 5 kicks. That is flippers kicks. Then you take photos on both sides of the transect. To take photos, a photographer should lay horizontally above the surface of the base of the ocean, and to be at least 0.7 to 1 meter, so that you can be able to capture a photo of 1 meter by 1 meter. And that's what we call photo quadrants. It's easy and it takes less time to take photos, but most of the work comes after the photo, which consists of annotating the photos using different annotation tools.
First of all, I'll define adult corals. Adult corals are the ones that have a diameter of about 10 centimeters and above. Those are the ones that we categorize them as adult corals. And to document adult corals, we normally use a transect line of 25 meters. So if you have a transect like this one, you measure 25 meters on the reef, and then you estimate 1 meter from the transect line. So the sample area is now 25 meter by 1 meter belt transect. All the corals that are within that transect line, we normally document 23 of the genera available because of the abundance in the Wayo region, and also they occur in different thermal ranges. So we always use the 23 genera to document adult corals. Once we've set up the belt transect, I use the 1 meter PVC pipe. This one helps me estimate the belt transect. So when I'm sliding or diving over the reef, this PVC pipe, which is 1 meter, helps me keep the range of 1 meter from the transect line. And this PVC also serves multiple purposes. I also use it for measuring the diameter of the coral. These yellow markings are different sizes, diameter. So you have, this one is 10 centimeter. Any coral that is beyond this range qualifies to be an adult coral. And then here is 20 centimeters. So this is another category. And then we have, here is 40 centimeters. So this is, when I put it on top of the coral, I can know which range of diameter the coral is. So once I've measured the diameter of the coral, I go into the slate and document which size category it belongs and which genus it is. Sometimes people record up to the lowest taxa possible, like species, but most of the time we need at the general level. And then, so along that 25 meter by 1 meter belt transect, I record all the 23 genera that occur there. So every coral colony that I encounter, I have to document its health condition. And we have different categories of health condition. The first one is whether it's healthy or partially dead. The second category is bleached. Is it bleached? Is it partially bleached? Or is it pale? The third category is if the colony has been predated upon by mostly fish. And the fourth category is whether it has diseases on it. Sometimes you can spot a disease on the coral. So once we complete the sampling the first belt transect, we have to do a second belt transect. With this kind of surveys, we have to do a minimum of 2 transects or belt transects. That's what qualifies the data for further analysis. Sometimes a coral can extend outside the belt transect. So you find that one bit is inside the belt transect, another bit is outside. So whenever a coral colony extends outside the belt transect, you have to estimate, does it, at the center of the coral, does it fall inside the belt transect? If it does, then you treat it as part of the corals to be sampled inside the belt transect. But if the center of the colony falls outside the belt transect and only a little bit of it is inside the belt transect, then you disqualify it. Does it fit to be sampled within that belt transect? So once you have sampled the first belt transect and then you are done with the second belt transect, you have enough data for analysis of the adult coral colonies in the reef.
So for the coral recruit survey, it's essentially in 2 steps. But first, what is a coral recruit? So a coral recruit is any coral that is smaller than 10 centimeters in diameter. To do the survey, you would first lay a transect of 25 meters along the reef. Then the second step would be to lay a 1x1 quadrat along that transect. Once that line is laying, you start using a quadrat, and we use a 1x1 meter quadrat. And this you would lay first at the start of the transect line, so at 0 meters. Then you would continue doing this every 5 meters. So you would move your quadrat along the transect line. So if you have a transect line of 25 meters, you would survey 6 quadrats in total. Now, within each quadrat, you're going to count all the coral recruits. And you would also divide them in size classes. We have a size class from 0 to 2.5 centimeters, 2.5 to 5, and 5 to 10 centimeters. And also, you would identify each coral to genus level, at least. So how you would really do that, if this quadrat is laying in the water, you just start at one point and you see if there's any small coral recruit. And if you see one, you would write down the genus and you would turf in which size class it is. And you would continue doing that until you've counted all coral recruits in your quadrat. Now, if there are many coral recruits in your survey, you could also add 2 lines in the middle of the quadrat to kind of divide it into 4 squares of 25 by 25 centimeters. And that way first start counting in one of those squares, then moving on to the next square. And that way it's easier to keep track of which corals you already counted and which ones not yet. After you finish this first transect, you would do one more in the same general area. So then in total you cover 2 transects. So in total 12 quadrats. So if you fully cover those 12 quadrats, you've got enough data to know the status of the coral recruits on that reef.
For fish abundance methodology, you need 2 observers, observer 1 and observer 2. Observer 1 will indicate to observer 2 where to place the transect for the start of the transect. This will be the deepest point of the dive for the first transect. Observer 2 will attach the transect to a rock, and then observer 1 will swim in front of observer 2 in a straight line. The transect tape is 50 meters long, and the area is 2.5 meters on either side of the transect tape, so 5 meters across and 50 meters long. When you reach the end of 50 meters, observer 2 will alert observer 1 that they've reached the end of the 50 meters and they stop the transect then. Observer 2 will then give the observer 1 the transect tape, and observer 2 will swim back along the transect tape while observer 1 reels it in. So on the way out, observer 1 will be doing the fish abundance and biomass of the majority of the families that we survey. On the return transect, observer 2 will only be doing the abundance and biomass of butterfly fish, angelfish, and small species of surgeon fish. The species that the families that observer 1 surveys on the outgoing transect are highly mobile and tend to move out the way, which is why the observer 1 goes first, followed by observer 2. On the return transect, observer 2 will do the small species that tend to be sedentary and don't move very much. These are butterfly fish, angelfish, and 4 small species of surgeon fish. When you're learning to estimate size classes, you can use model fish you know the size of the fish, and you practice this underwater with scuba with a buddy. So we've got different models of different sizes, and we know what size each of these is. So you write on the back what the size is, so the person who's learning, who's practicing, doesn't know the size of the fish, but the buddy will know.
So the data that is collected from the coral reef monitoring programs belongs to the primary data collectors. So in some cases that's us when we go out monitoring coral reefs, but usually we're doing it with partners, and partners will do it themselves. So, and each data collection team will use the data for their specific needs, whether that's managing a marine protected area or understanding the trends of what's happening in their area. And we try and help various partners understand how they can analyze the data and how it can support decision making. But then what we also do is we integrate or we combine data at higher levels. So we work with local government entities, for example, here in Kenya, the counties, to compile data across coral reefs within the counties and write reports, or help them write reports on the status of coral reefs in the county. We also aggregate data to national levels to feed into national reporting, and then also at regional levels for regional reporting within the Eastern African region on the status of reefs, and then of course at global levels as well. And so at all of these different levels the data can be used and aggregated, and hence the importance of the standardization, so that we can get the same information being used at all levels for consistency in decisions and policies to protect the reef.
So far we are now trying to use some of this data, for example, under the IUCN Red Listing of Ecosystems. We've been working with that with CODIO also and other partners in the country, Chemfree and other organizations to identify the important areas based on the data that we've collected so far. That information has guided us a lot in trying to appreciate that and also identify the important area for certain species. And also in some areas we've also recommended closures for communities for conservation of certain species, or maybe to enhance fisheries productivity.
My hopes for people using this manual is I'm going to focus on the fish because they've been more poorly monitored over the years. There's been much less consistency, less regular monitoring. So I'm really hoping it's going to encourage people in the different countries of the WIO to use some standards, monitor these families, and then we're going to have much better data on the state of fish populations. And I'm also hoping the protocol's really going to encourage people to go beyond the family level. Family level is okay, it's a butterfly fish or it's a snapper. But actually, there's much more interesting information if you get it at species level. Pick some families and learn the species in those families, because they'll tell you a lot and they'll give you a much more nuanced indication of how is that reef responding, A, to climate change, but B, to whatever management that community or that country is putting in place.
So we hope that this manual and this video can really ramp up the training that is available across the region. A video like this can be used by anybody, anywhere, and can really help programs to build up their own capacity without necessarily needing instructors to come in from outside. So we hope it gets used as widely as possible throughout East Africa and of course anywhere else in the world.
I'd like to say for people monitoring coral reefs outside the WIO, i.e. in the rest of the world, that our protocol is completely compatible with the global coral reef monitoring GCRMN process. It's therefore highly relevant to people working in the Pacific, anywhere in the Pacific or the Eastern Indian Ocean or up in the Red Sea. Species might differ a bit, but the standard protocol is the same, and therefore it's valuable for everyone. We need to be monitoring all around the world on coral reefs.